So our third speaker for this session is Liz Speliotes. Liz is an associate professor of internal medicine and computational medicine, bioinformatics, interested in non-alcoholic fatty liver disease, obesity, and related metabolic traits. And Liz will today be talking about using MGI and Biobank data to inform precision medicine. Thanks for being here.
Great. Thank you very much for having me. I really appreciate the invitation. So as Mike mentioned, the main interest in my own group is on obesity and non-alcoholic fatty liver disease. And the reason why we're interested in this is that they're very common diseases that probably will affect either you or someone in your family. For example, in the United States, more than 66% of individuals are overweight and 33% are obese. Obesity affects more than 1 billion people worldwide out of about 6 billion people. It accounts about 17% of our healthcare costs. And then we study various types of obesity. So overall obesity can be measured by something that's very commonly measured in EHRs, such as the MGI, which is body mass index, which is measured by someone's height and weight. But we also do study sort of redistribution of fat within the body. So sometimes, some people can get more abdominal obesity called a higher waist to hip ratio. They can get more lower extremity obesity, or they can get something called lipodystrophy, where you have abnormal sort of accumulation of lipids. So we study that whole range of phenotypes. And then one of the places where fat can deposit is also in the liver. And when it deposits in the liver, you actually get a liver disease. Shown here is sort of a normal liver, but then as people gain weight, you can deposit fat in the hepatocytes, and this can lead to inflammation and scarring and ultimately cirrhosis and even predispose to cancers. And this is the whole range of something called non-alcoholic fatty liver disease. And this has also increased with the rise of obesity, and now it's becoming really the prevalent disease of the liver, accounting for about 24% in global prevalence. And most of these diseases, both obesity and non-alcoholic fatty liver disease, we really have a very poor understanding of what's causing these because they're relatively new kind of phenomenon in our society. And therefore, we have no effective medical treatments, which is one of the reasons why I'm very interested in it. It's also a very big unmet medical need that we're trying to fill the gap on. And then the other thing that we're very interested in is that, even though I tell you that these traits are correlated, people that gain weight get more fatty liver, but these really are not perfect correlations. So here, as I told you, this is a normal liver. As people gain weight, about 90% of them will deposit fat in their liver. But even in bariatric patients with BMIs in the 50, 10% of them actually don't have fat in their liver. So there is genetic variation in how people distribute fat. And we think about 20% of these people that get fat in their liver go on to get inflammation around that fat and get something called non-alcoholic steatohepatitis. And about 20% go on to get fibrosis or cirrhosis. And then in work that I did in the Framingham Heart Study, we showed that individuals that have more than 30% fat in their liver, 66% of them had criteria for having this metabolic syndrome, which is having 3 or more of the following, which is central obesity, high triglycerides, low HDLs, impaired fasting glucose or diabetes, and high blood pressure. And that some of these individuals can go on to get cardiovascular disease. And in fact, with a fellow here, we went on to show that some individuals with fat in their liver actually can more directly predispose to cardiovascular disease, even independent of some of the metabolic syndrome traits. So one of the key things that we're interested in my group is, why do some people deposit fat and then go on to get liver disease? Why do some of them get metabolic syndrome? And can we use genetics to really identify who's going to go in what direction so that we can give them better precision care? Because unfortunately what happens right now in the clinic is that you'll show up, you'll say, oh, you're obese, okay, you need to diet and exercise. Well, maybe that's helpful for you, but chances are that's not going to be helpful for everybody, because some of those people are at high risk of these diseases and these complications, but some of them are not. Some of them are going to be sort of a healthy obesity. And right now you don't know who you're talking to in front of you, so you tell them to do these things and you say, well, how bad could that be? We just told them to diet and exercise. Everyone should do that, right? Well, there's a cost to everything. So, there's a cost to diet, there's a cost to joining a gym, there's time spent, there's stress, right? You're just telling someone to do something stressful. And then the other thing that's happening in medicine now is we're just throwing medicines at people, right? So we're just like, this is a proton pump inhibitor. It's not a lot of side effects. Go on this, go on an aspirin, go on these things. But as we're sort of looking at that more closely, what we're discovering is that these things are not benign. They can cause side effects, and even a 5 to 10% rate of side effects or nuisance or other things can cause problems. And that's really where the hope of precision medicine is, to better define sort of your risk and kind of tailor the recommendations to what you really are susceptible for, as opposed to what the general population is susceptible for. And of course, I believe in the power of genetics, and the reason why I believe this is if you can find genetic variants that predispose to any trait, by definition it's in the causal pathway. And that's really important in medicine, because in medicine for a long time we've gone after things that are correlated but not causal, right? So money and power are correlated, but you don't know if the money caused the power or the power caused the money. Same thing in medicine. You have these inflammatory markers that are fine, obesity. We've gone after them, but actually they're the cause from obesity. They're not actually, they're the result of obesity and not the cause. And so there's a lot of money being spent putting people on medications that are really not going to work, going after targets that are not really going to affect the actual course of the person's disease. And so if you can find genetic variants that associate with any trait, you can use them to understand causal influences to that trait, understand your individual susceptibility to it, define personal risk, and develop tailored therapeutics. And in fact, we, along with many colleagues and collaborators here, have gone on to carry out some of the biggest genome-wide association studies for measures of obesity, like body mass index, which is shown here, waist-to-hip ratio, fatty liver disease, liver function tests. And it's been a very rewarding experience, because now we're beginning to sort of see the causal landscape of these diseases that we really don't understand. So that's extremely exciting to us. The other things is that these are complex genetic traits. So as this audience knows, you know, it's not a gene causes these, but it's really the genes in combination with the environment that then goes on to predispose to these. And we've done heritability estimates about how heritable obesity and NAFLD is, and it does suggest that at the population level we can study these. And then we're not just interested in just the genetic factors. I think it's for sure for something like fatty liver disease, which is only about 30% heritable, that the environment is going to play a big role in this. And so we are also interested in identifying environmental factors that interact with these genes to multiplicatively sort of enhance disease. And the reason why that is, you know, you may not be able to change all the 50 things in your genome that are causing you disease, especially for these polygenic diseases. But if you can find sort of environmental variables that multiplicatively influence your disease risk, what that means is that if you're at risk of a disease and you interact with the right environment, like high insulin or low insulin, you have an multiplicative increased risk if you have high insulin of getting fatty liver disease. But if you change that, if you go from a high insulin state to a low insulin state, the diseased individuals actually have a better outcome. They actually gain more, right? So if this was exercise, this person would gain more by exercising than the other person that didn't exercise. And that's exactly what we're trying to get to. We're trying to say, okay, for you, you're at super high risk of this, so you should focus on these 3 things, because otherwise you just can't focus on everything. You know, you're all predisposed to a lot of things, but you just can't be doing everything for everyone. And so shown here, for example, is one of our recent papers where we look to see if genetic variants that predispose to fatty liver disease interact with certain serum traits and other measures, and we did this in a large data set that we had. And some of the interesting things that we found is that we found that insulin, glucose, triglycerides, and BMI all interacted with this genotype to cause fatty liver disease. But when you do conditional analyses, it actually turns out to be the insulin resistance that's really the most causal. And that was a little bit surprising, in that we've known for a long time that diabetics are predisposed to getting fatty liver disease. And most people think that, oh, diabetes is defined by glucose, so therefore high glucose is really what's causing this. But it's probably not the high glucose. It's probably something in the insulin resistance that changes a use in nutrients that is causing the fatty liver. I mean, we have some hypotheses, like, for example, you have glucose, you have insulin, you can't get that into your cells, and so what happens is actually you have a lot of fatty acid release from the tissues that happens, and that actually may be more what's coming to the liver and causing the fatty liver than necessarily the glucose part of it. So these are clear hypotheses that you can now test and bring to the clinic, where you can then in the future have a more directed approach to interfering with the environmental variable that you identify for this disease. So then the other thing that we do is PHWAS and pleiotropy effects. And in our original papers, we had variants that predispose to fatty liver and also fibrosis, but we also looked to see what kind of effects they have on other serum traits. And in fatty liver, epidemiologically, you see increases in LDL, lower HDLs, higher triglycerides, and higher fasting glucose and insulin resistance. But really, we didn't see this pattern across really any of our variants, which was a little surprising. But what this tells you is that not all fatty liver is the same, right? You can see that there's different patterns of effects, and some of these patients that have fatty liver don't really have a lot of effects on other traits. And some of them actually have effects that are opposite of what you see epidemiologically, and this is really the basis of precision medicine. There are subgroups, and right now we're calling something fatty liver, but there's probably subgroups of fatty liver, just like there's subgroups of diabetes and subgroups of obesity. And eventually, if you can find these and define these subgroups, you can then now, with genetics, link them to targets that you can use for therapeutic intervention for more precise treatments. And in addition to this, because of this kind of precision, we now have cell line models based on some of these gene targets where we can knock down genes, overexpress them, and we can try and match them to particular drugs so that now we can get particular drugs that target these subclasses. And so in the future, you can have more on-target effects and fewer side effects, which is what is shown here when you take a medication. Oh, wow, okay, that went fast. All right, so the other thing that we're doing is we're enabling the genetics and genomics, and we're trying to use it to study many other diseases like diverticulosis, diverticulitis, cirrhosis, alcoholic liver disease, inflammatory bowel disease, lipodystrophy, and laminopathies. And really for MGI, the traits that we've looked at the most are things like fatty liver disease, cirrhosis, liver function test, height, weight, body mass index, diverticulosis, and then we're going to use MGI for more interventions by genotype and IBD. And some of the lessons that we've learned in looking at some of the MGI data is you definitely need to clean the data. I mean, there's definitely many clerical errors like BMIs of 900 that just do not make any sense. The other thing is when patients are hospitalized, their serum values can be very different than their baseline. So if we have longitudinal data, we usually eliminate all the hospitalization values, and we try to use the median values per person if we have multiple to try and get rid of these outliers and some of these errors. The other thing about these data is that the searches are limited to what's known and documented. So clearly, if you're trying to make new discoveries or new subtypes, nobody's going to document that, right? They're just clicking off what is already in their drop-down menu. So just keep that as in the back of your mind about this kind of data. And then a lot of our diagnoses are present in these databases, a lot more than you'd see in the average population because of likely we're tertiary care centers, so more sick people come here. And then we've mostly used this for replication because some of our other data sets are very large, so we use this mostly for verifying SNPs once we have a candidate list versus GWAS discovery. And we found, on the genotyping side, that it's been most helpful for verifying common variants, occasionally for low frequency variants. And so far for our rare variants, it's a touch and go, but most of the time we don't see them there. And then, again, this is also limited to European ancestry for the most part. It's powered for European ancestry, so our ancestry-specific effects we don't use it much for. And then we have found that, for our phenotypes like fatty liver disease and cirrhosis, these are underdiagnosed anyway and misdiagnosed often. So we have found that, if you do chart reviews, about 30% of the cases in these diagnoses are probably miscoded or misidentified. And then we have used natural language processing to look at pathology reports and also to look at radiology reports, and we do increase the number of cases by about 15% when we do them. So those are things that I think may be useful to you if you're using this data set. So how have we used these, for example? So there's a lipodystrophy genetic score that's been published, and one of our fellows sort of used this to see if the number of alleles gives you a higher rate of fatty liver disease or cirrhosis, and comparing the top 10% versus the bottom 10% of scores. And he did see that there was an increased risk of fatty liver, about 36% of top versus bottom and 22% in the MGI for cirrhosis versus non-cirrhotics. We've also used it to replicate variants that were associated with cirrhosis from the UK Biobank, and shown here we were able to replicate a lot of these common variants. One of these common variants was an indel, which is actually not well imputed in the MGI, so we didn't have that data. But the other ones, we were able to replicate some of these in MGI, despite the much smaller size. I told you about the gene-environment interactions, so we've actually designed an intervention by genotype trial with one of the people at the School of Nursing. And what we're going to be doing there is, again, taking the risk individuals that have GG from Michigan Genomics Initiative, and we identified 116 of these in the older data set that also have high ALTs. And then what we're going to do is put them on a low carbohydrate diet that actually reduces insulin resistance, and this is a very effective diet for reducing that insulin gradient that I told you about. And we're going to be determining their liver fat or fibrosis at 4 months to see if we can actually have that higher effect in the risk genotype versus the non-risk genotype. So you heard from Lily about how she's used it to, again, replicate variants that are associated with diverticulosis in UK Biobank in Michigan Genomics, and we're doing polygenic scores and gene bioenvironment interactions for this trait. And then Kelly Cushing is another one of our faculty that we're working with to look at IBD. So IBD patients have an increased risk of skin cancer, and this is thought to mostly be due to medication use, but we noticed when we did a FEWAS in the UK Biobank that some of the variants really associated with non-melanoma skin cancer, and then we did regression analysis to see whether or not these variants, independent of any medication use, had an effect on IBD, and the answer was yes. For at least 4 out of 5 of these, they did have independent effects. And so we are looking to see if we can now replicate this in MGI, where actually we have more IBD patients than you do in UK Biobank, because it's a referral center for that. And also we have very good medication use and much better data that she is going to curate in MGI. And then Graham Brady is another one of our fellows, and he's interested in lamin proteins. So these are rare variants in lamins can cause some progeria and other multi-system syndromes. And then we found that some of his low-frequency variants had effect on fatty liver in our consortium data, and he's looking to replicate those now in UK Biobank and the MGI data, and he's going to make cell line and zebrafish models of that. So overall, the MGI has been a great resource, and then other hopes that we have for it is actually a link to pathology data so that we can actually do like gene expression studies or link them to other histologic markers of disease once we identify sort of a new association, link to imaging, and link to prescriptions, exome sequencing, more diverse population, and integration with other biobank data would be very helpful to our research program to expand the use of this. So I'd like to thank everyone in the MGI for all their wonderful efforts. This is a great data set that's been very useful to us, our collaborators, and my lab. Thanks.
Thank you, Liz. Great talk. I was wondering, because there are some studies that have come out which have shown that the knowledge of the polygenic risk score has really influenced behavior changes, even if you don't stratify with respect to a certain genotype. Mm-hmm. But just knowing that you're in the highest decile, mm-hmm. Is there a way to incorporate those kind of changes in your trial and in your study?
Yeah, no, I mean, I think, so actually Laura Saslow is a psychologist. So we are, in addition to our intervention and measuring the fatty liver, so she's getting a very detailed questionnaire about, what your eating habits are like, what your stress level is like. So certainly we could add things like, if you knew about this, would it change your behavior? And I'm hoping it's a positive trial in that the people that are highest risk actually have the most benefit, in which case, hopefully they'll say that it was useful to them, going to an intervention or not. But we can certainly add that to our questionnaire. All of our stuff is online, so adding sort of other components to it to evaluate how much knowing about their genetic risk adds to their behavioral change may be interesting to study. We'll add that to our IRB.
Exactly. So. When you were looking at your environmental interaction, you seem to be mostly talking about other measurements from the individual. Mm-hmm. Have you considered real environmental facts like access to fresh food or something like that?
Yeah, so actually Vince is looking at that right now, so that's his K word. So I'm trying to get him so we're looking at that in UK Biobank, so they have a lot of variables collected in terms of dietary intake. And we're looking at that data right now to see if we can actually link it to, gene-environment interactions with specific, intake. And then we've actually gotten more and more data about, total calories per day or other things that we can more specifically tailor to in the diet. I can tell you that I also have another study in China And there, we did have also a very good dietary intake where we created our own FFQ and 24-hour recalls, and we found associations with fatty liver with, red meat intake, and it's actually also come up in UK Biobank, and so there may be something about, things that I wouldn't have necessarily sort of thought, and I would have said, oh, maybe it's like alcohol or something else, but, over time, we're controlling for alcohol, we're controlling for these things, and so, unless there are confounders in every single population, it seems to be coming up, and so it's a little bit unexpected. I don't know if it's heme or something in there that somehow interacts with weight and other things to predispose to liver disease, but that may be real. And so I think we just need to do more of these things, and especially if we can replicate it across multiple biobanks and multiple data sets, it may not be that there's, bias or this or underreporting of alcohol or something that I initially thought, it's just, people that eat meat have more money and therefore they're going out and drinking wine and doing other things. But even though we controlled for it. So those are very interesting things that we can then, tailor people's diets to that over time.
Thank Liz and all three speakers for